because like I said, Ms. Dorado is coming at the end of class. But organ donation. For most organs, a person has to sign up to be an organ donor. Most of the time, people have a little pink square on their driver's licenses that say donor, just in case they get in a car accident. So the doctors know that that person was an organ donor. But then for most organs, they have to die. You can't donate your heart to someone and still live yourself. So that is the sad reality of organ donation. There are some organs like kidneys and even part of your liver that you can live without. You can live with only one kidney. You can live with part of a liver. But most of the time, if you are an organ donor, that means you have to die to be able to donate your organs, which is awful. So scientists are working on a different way. Has anyone heard of any alternatives to organ donation? We'll get to it. I'm just curious if anybody's heard anything. Okay. Our biggest hurdle to organ donation is our own immune system. So if we need a heart transplant, somebody dies, they're what's called a match for us. We'll talk about matching in a little bit. We get their hearts. Their heart is still a foreign substance to our bodies. So doctors have made medication to basically suppress our immune system so we can keep that heart or lungs or whatever else. So we have 3 levels of defense against the immune system. We're going to talk about the whole immune system first, and then we'll talk about organ donation and how it relates, and a couple other things. So in the course of an average day, we encounter many different pathogens. Pathogens are disease-causing agents, viruses, bacteria, fungi, parasites. Fortunately, we possess several levels of defense against infection. And this is a link on pathogens to the coronavirus. The coronavirus is the biggest pathogen that is affecting us right now. We've heard a lot about it. We've talked a lot about it. Your discussion this week, or your assignment this week, is reading an article about it. So you guys are going to learn about that. I'm not going to show that video right now. If you want to learn more about the coronavirus, feel free. Our skin is our first line of defense. It is a physical barrier between us and anything that wants to get in. On our skin, we have in our skin, we have mucus. That's part of our mucus in our nose and our mouth is to trap pathogens and kind of get them out before they go any further and make us sick. We have lysozyme, which is kind of like an acidic substance in our sweat. Our sweat can be acidic, kills bacteria. The next level is called the innate immune system. These are non-specific responses, so they will kill everything in its path. Inflammation, and then something called the complement system. Inflammation is when, if we get a cut and it gets red, that redness is called inflammation, or even when we get a fever. When we get a fever, it's our body raising the temperature of our blood, because many bacteria don't like hotter blood. They like it in our normal body temperature. And then our third level of defense is called the adaptive immune system. It's very specific responses. There's pathogen recognition and neutralization. Our cells remember the pathogens we have encountered in our lives. And the more often we encounter it, the quicker our immune system mounts a response. There's something called antibody-mediated immunity. Our body produces antibodies, and then cell-mediated immunity that we'll talk about. And each level initiates an increasingly aggressive response. So it starts out kind of light. We're just trying to block the pathogens and keep them out. And then they get really specific and really aggressive, where our cells basically just eat the bacteria and destroy it. Inflammatory response is a general non-specific response to a foreign substance in our bodies. All of our cells have ID tags. We know what's supposed to be in us. We know what our own cells look like. So anything outside of that is considered a foreign substance, and our body attacks it. And there's stages. Invading organisms penetrate the skin or epithelial cells. Epithelial cells you have in your cheeks or other cavities. Macrophages engulf and destroy bacteria, secrete something called cytokines. You have mast cells that secrete histamine to prevent an allergic reaction. Cytokine and histamine dilate capillaries to increase blood flow. To dilate means to make bigger, so more blood goes through faster. And then neutrophils migrate to the infection site and engulf bacteria. Clotting factors seal off infection area. Phagocytes clean up debris from dead cells. These are all different white blood cells that have different functions. Genetic disorders can cause inflammatory diseases. Inflammatory bowel disease, Crohn's disease, ulcerative colitis. These are all disorders that produce extra inflammation in the body. The complement system participates in both nonspecific and specific immune responses. And there are stages to this system. Complement proteins are activated at the site of infection. There's a cascade of activated complement proteins. It's kind of like a domino effect. That's what a cascade is. You have one thing gets activated, that activates the next thing, then the next thing, then the next thing, then the next thing. There's a formation of membrane attack complexes called MACs for short. And the end result is lysis of the pathogen. Lysis basically means it gets blown up. It has to die. And then there's a whole video on the complement system. No, I'm not going to watch that right now. Okay. And the adaptive immune system is the last line of defense against pathogens. Chemical and cellular reactions neutralize and destroy a pathogen. There's a memory component. Your cells remember which pathogens your body has been exposed to. It allows a rapid response to the second exposure. That's what people are trying to figure out right now about coronavirus victims. They now have cells in their body called antibodies that remember the coronavirus. So if they get it a second time around, their body should react faster and kill it faster, make it go away faster. Adaptive immunity has two interconnected systems: antibody-mediated immunity, regulated by B cells, and helper T cells. Those are both white blood cells, just different kinds. And cell-mediated immunity, controlled by cytotoxic or killer T cells. It's directed against cells infected by pathogens, cancerous cells, and transplanted cells. So our killer T cells will kill our own cells if they're infected or something is wrong with them. And that's what we want. We don't want our infected cells to spread. The adaptive immune response is mediated by white blood cells called lymphocytes. They're formed in the bone marrow. These are the B cells. B cells are a specific kind of lymphocyte. Lymphocyte is a fancy name for white blood cell. The B cells produce antibodies. The antibodies get displayed on the cell surface. They're the little Y-shaped things in the picture. Antibodies bind to antigens on a pathogen cell surface. So bacteria has little things called antigens on its surface. And the antibodies recognize the antigens. Each B cell produces only one type of antibody. So you have hundreds of different B cells in your body for each type of antigen, each type of pathogen your body has encountered. So everybody's encountered a cold or the flu. Some people have had pneumonia or maybe mono. And each one of those sicknesses now has antibodies flowing through your blood, just in case your body sees them again. T cells is another type of lymphocyte or white blood cell. They migrate to the thymus as immature cells. Your thymus is an organ that kind of covers your throat inside. It's a butterfly-shaped organ. And then when they're mature, they circulate and they can be found in the spleen and lymph nodes. Your spleen is in your abdominal area, and your lymph nodes are when you're sick and doctors are feeling your throat like this, they're feeling for your lymph nodes. Because if they're swollen, that means they're working really hard to get rid of whatever you have. T cells produce T cell receptors, which are just little structures on the surface of T cells. And the TCRs bind to pathogen proteins on the surface of infected cells. Like I said, the T cells attack our own cells if they are infected. Clonal selection. An antigen binds to a specific antibody on a B cell, called a naive B cell because it's not previously encountered an antigen, so it's not specific yet. This triggers mitosis and the buildup of a large population of cells derived from that activated cells. You can see in the picture the antigens in red. The antigen binds to an antibody specific to it on a naive B cell. So the antibody has to be the right shape. And then B2 gets activated and it gets cloned. Mitosis just makes a bunch of them to be specific to the sickness that is infecting us. MHC is called the Major Histocompatibility Complex. It's a set of 140 genes on chromosome 6. It encodes recognition proteins that prevent the immune system from attacking its own cells. So like I said, our own cells have little ID tags on them, so our body knows this should be here. Don't attack it. And this is coded for by chromosome 6 and the MHC. The human leukocyte antigen genes is a set of 9 genes within the MHC. So the MHC on chromosome 6 is this big. Then you have 9 genes within that called the HLA. Each gene has many different alleles, allows for countless allele combinations or haplotypes. And the HLA plays a big role in organ transplantation. that set of 9 genes has to be compatible with another person in order to accept their organ if you need it. The immune system has 2 interconnected parts, like we talked about: antibody-mediated immunity, regulated by B cell antibody production, and cell-mediated immunity, controlled by T cells. And the 2 systems are connected by helper T cells. So there's a lot of different cells that you have to know about to understand the stages of the immune response. And we are going to watch a video overview to connect all these cells to hopefully make it a little clearer. The steps involved in the responses of the 2 systems are similar. B or T cells recognize an antigen. The cells become activated and divide to form clones. The clones attack and destroy the invading pathogens, which will clear the antigens from the body. And then some of the activated cells form memory cells that circulate through the body, ready to mount a rapid and massive response if the same pathogen invades the body again. Here's the steps and the overview of it. This video we are going to watch because this is an overview.
Every second of your life, you are under attack. Billions of bacteria, viruses, and fungi are trying to make you their home. So our bodies have developed a super complex little army with guards, soldiers, intelligence, weapons factories, and communicators to protect you from, well, dying. For this video, let's assume the immune system has 12 different jobs. For example, kill enemies, communicate, etc. And it has 21 different cells and 2 protein forces. These cells have up to 4 different jobs. Let's assign them. Here are the interactions. Now, let's make this understandable. First of all, let's add colors to the jobs. Now, let's illustrate the cells. The central color represents the main job of the cell, while the surrounding ones represent secondary duties. Now the immune system looks like this. Now the interactions. Isn't this complexity just awesome? For this video, we will only talk about these cells and ignore the rest. So what happens in the case of an infection? It's a beautiful day when suddenly a wild rusty nail appears and you cut yourself. The first barrier of the immune system is breached, your skin. Nearby bacteria seize on the opportunity and enter your wound. They start using up the body's resources and double their numbers about every 20 minutes. At first, they fly under the radar, but when a certain bacteria population is reached, they change their behavior and start to damage the body by changing the environment around them. The immune system has to stop them as fast as possible. First of all, your guard cells, known as macrophages, intervene. They are huge cells that guard every border region of the body. Most of the time, they alone can suffocate an attack because they can devour up to 100 intruders each. They swallow the intruder whole and trap it inside a membrane. Then the enemy gets broken down by enzymes and is killed. On top of that, they cause inflammation by ordering the blood vessels to release water into the battlefield, so fighting becomes easier. You notice this as a very mild swelling. When the macrophages fight for too long, they call in heavy backup by releasing messenger proteins that communicate location and urgency. Neutrophils leave their patrol routes in the blood and move to the battlefield. The neutrophils fight so furiously that they kill healthy cells in the process. On top of that, they generate barriers that trap and kill the bacteria. They are indeed so deadly that they evolved to commit suicide after 5 days to prevent them from causing too much damage. If this is not enough to stop the invasion, the brain of the immune system kicks in. the dendritic cell gets active. It reacts to the signals of the soldiers and starts collecting samples from the enemies. They rip them into pieces and present the parts on their outer layer. Now, the dendritic cell makes a crucial decision. Should they call for antivirus forces that eradicate infected body cells or an army of bacteria killers? In this case, antibacteria forces are necessary. It then travels to the closest lymph node in about a day. Here, billions of helper and killer T cells are waiting to be activated. When T cells are born, they go through a difficult and complicated training process, and only a quarter survives. The surviving cells are equipped with a specific setup. And the dendritic cell is on its way looking for a helper T cell with a setup that's just right. It's looking for a helper T cell that can bind to the parts of the intruders which the dendritic cell has presented on its membrane. When it finally finds one, a chain reaction takes place. The helper T cell is activated. It quickly duplicates thousands of times. Some become memory T cells that stay in the lymph node and will make you practically immune against this enemy. Some travel to the field of battle to help out. And the third group goes on to travel to the center of the lymph node to activate a very powerful weapons factory. Like the T cells, they're born with a specific setup. And when a B cell and a T cell with the same setup meet, hell breaks loose. The B cell duplicates rapidly and starts producing millions of little weapons. They work so hard that they would literally die from exhaustion very fast. Here, helper T cells play another important role. They stimulate the hard-working factories and tell them, "Don't die yet, we still need you. Keep going." This also ensures that the factories die if the infection is over, so the body doesn't waste energy or hurt itself. But what is produced by the B cells? You've heard of them, of course, antibodies. Little proteins that are engineered to bind to the surface of the specific intruder. There are even different kinds of antibodies that have slightly different jobs. The helper T cells tell the plasma cells which type is needed the most in this particular invasion. Millions of them flood the blood and saturate the body. Meanwhile, at the site of infection, the situation is getting dire. The intruders have multiplied in number and start hurting the body. Guard and attack cells fight hard, but also die in the process. Helper T cells support them by ordering them to be more aggressive and to stay alive longer. But without help, they can't overwhelm the bacteria. But now the second line of defense arrives. Billions of antibodies flood the battlefield and disable lots of the intruders, rendering them helpless or killing them in the process. They also stun the bacteria and make them an easy target. Their back is built to connect to killer cells, so they can connect and kill the enemy more easily. Macrophages are especially good at gnoming up the bacteria which antibodies have attached to. Now, the balance shifts. In a team effort, the infection is wiped out. At this point, millions of body cells have already died. No big deal, the losses are quickly replenished. Most immune cells are now useless, and without the constant signals, they commit suicide, so as not to waste any resources. But some stay behind, the memory cells. If this enemy is encountered ever again in the future, they will be ready for it and probably kill it before you even notice. This was a very simplified explanation of parts of the immune system at work. Can you imagine how complex this system is? Even at this level, when we ignore so many players and all the chemistry. Life is awfully complicated, but if we take the time to understand it, we always encounter endless wonders and great beauty.
All right.
Every second of your life, you are under...
Go. So antibodies, molecular weapons against antigens. Antibodies secreted by plasma cells are Y-shaped protein molecules that bind to specific antigens in a lock and key manner to form an antigen-antibody complex. Antibodies belong to a class of proteins known as immunoglobulins, or Ig for short. And there's 5 different classes with different structure, size, and function. Like he was saying in the video. As antibody structure, you got the Y shape, you got antigen binding sites, that's the specific area that gets bound to an antigen. And there's different regions, different chains. And then there's all the Ig classes, IgD, M, G, A, E. Some of them you get naturally, some of them, if you're breastfed, you get from your mother, some of them you get from vaccines. The cell-mediated immune response is the formation of antigen-presenting cells, so that one cell that took in the bacteria and then presented pieces of the bacteria on its surface is called an antigen-presenting cell. Antigens on the APC bind to this TCR, the T cell receptor on a CD8 cell. CD8 cell is a type of helper T cell. And then you get the clonal expansion. Once that cell is activated, it goes through mitosis and produces millions and millions of copies of itself into cytotoxic and memory T cells. The cytotoxic T cells will attach to the infected cell and secrete perforins. Perforins punch holes in the bacteria so it can get destroyed, so it dies. And then debris from dead cells is removed by phagocytes. There's a couple different videos that you can watch.
Activation.
No, we're not going to watch that right now. Immune system memory allows for a much more rapid response to the second exposure, and it's the basis for vaccines. And we're going to watch this video. It's going to go over vaccines in general, and then it's going to go over the COVID-19 vaccine that companies are working on and getting distributed right now so we can come back to school.
Vaccines prepare the immune system, getting it ready to fight disease-causing organisms called pathogens. A vaccine is introduced to the body to mimic infection, triggering the body to produce antibodies against the pathogen, but without causing the illness. Conventional vaccines usually contain a weakened or inactivated pathogen, or a piece of a protein produced by the pathogen called an antigen. RNA vaccines are a new generation of vaccines. Instead of the antigen itself, RNA vaccines contain a messenger RNA, mRNA, that encodes for the antigen. Once inside the body's cells, the mRNA is translated into protein, the antigen, by the same process the cells use to make their own proteins. The antigen is then displayed on the cell surface where it is recognized by the immune system. From here, the sequence of events is similar to that of a conventional vaccine. Some RNA vaccines also contain additional mRNA coding for an enzyme, which, after being translated in host cells, can generate multiple copies of the antigen encoding mRNA. This essentially amplifies the production of antigen from a small amount of vaccine, making the vaccine more effective. These are called self-amplifying RNA vaccines. RNA vaccines are easier and safer to produce than conventional vaccines. This is because mRNA molecules can be synthesized in a cell-free system using a DNA template with a sequence of the pathogen. While conventional vaccines usually require a more complicated and risk-prone process of growing large amounts of infectious pathogens in chicken eggs or other mammalian cells. Without the risks of being contaminated by infectious elements or allergens from egg cultures, RNA vaccines are also safer for patients. Because protein synthesis occurs in the cytoplasm, RNA molecules do not need to enter the nucleus, so the possibility of them integrating into the host cell genome is low. RNA strands are usually degraded by cellular enzymes once the protein is made. The relative simplicity of the production process makes it easier to standardize and scale, enabling rapid responses to emerging pandemics. Other advantages include lower production costs and the ease of tweaking RNA sequences to adapt to rapidly mutating pathogens. On the minus side, it can be challenging to deliver mRNA effectively to the cells, since RNA sequences and secondary structures may be recognized and destroyed by the innate immune system as soon as they are administered intravenously. These limitations can be overcome by optimizing codons, using modified nucleosides to avoid recognition, and packaging RNA into protective nanoparticles. Another disadvantage is that most RNA vaccines require uninterrupted refrigeration for transportation and storage, which can be a hurdle for vaccine distribution. Research is ongoing to engineer thermostable vaccines. Vaccines.
So that's the latest on the COVID vaccine. It's an mRNA vaccine and it has to be kept cold, which is one of the disadvantages. One of them has to be kept like below freezing to transport it, which is really hard. Blood typing is based on the presence or absence of certain proteins on the surface of red blood cells. So this might get a little confusing because our red blood cells, those proteins are called antigens. Usually antigens are found on bacteria and viruses and foreign substances, but our red blood cells have antigens, certain ones. There's two blood groups of major significance. There's the ABO system and then the RH blood group. If anyone knows their blood type, mine is O positive. Positive means I have the RH antigen on my blood cells. Individuals produce antibodies against antigens that are not present on the surface of their red blood cells. So example, an individual with blood type A will produce antibodies against the B antigen. So this is why for blood donation, you have to know your blood type. If you're blood type A and you get injected with blood type B, your body is going to make a lot of antibodies against that B to kill it and get it out of its system because it sees it as a foreign substance. And there's been a recent study, an association between ABO and RH blood groups and COVID. In Canada, there was, and that's a link to the article if you want to read it, but in Canada, they found that people with blood type O are 12% less likely to become severely infected with COVID, which is interesting to me because I'm a science geek. There's a video on different blood types that will explain them all, basis for blood type matching.
Different folks may.
Organ transplants must also be immunologically matched. So not only does the person have to have the same blood type, they have to have the same HLA on their chromosome number 6. So successful organ transplants depend on HLA matching between donor and recipient. Recipient is the person that receives the organ. Mismatched HLA haplotypes lead to immune rejection. Closely matched HLA haplotypes still require immune suppression in recipients. And the best chance for a match is between related individuals, specifically siblings or parents. Identical twins are the best match because they have identical chromosomes. Their HLA will always match. There are two problems related to the immune rejection of xenotransplant. So xenotransplant, we are going to watch for a minute. I don't know why I went that way, but okay.
With sweet love and devotion, gently touching my emotion. How sweet it is to be loved by you. There are
are thousands of people who owe their lives to pieces of dead pig tissue. And no, I'm not talking about the admittedly life-affirming taste of bacon. For the last 50 years, we've been replacing damaged human heart valves with pig valves. So if we can transplant heart valves from pigs, why not entire hearts or kidneys or livers or lungs? How come you can walk around with a pig's heart valve inside of you, but you reject a kidney from another human? And out of all the creatures in the world, why pigs? So many questions. Transplantation of organs from one species to another is known as xenotransplantation, and it turns out to be pretty bizarre. But since there's a shortage of human organs, figuring out how to get a steady supply of animal organs and tissues that we could use is something we might want to figure out. Transplantation from human to human is tricky enough, much more difficult than matching blood types. The new organ is often rejected by the recipient because of a protein called human leukocyte antigen, or HLA. Almost every type of human cell has different kinds of these proteins on its surface, and normally they're the immune system's undercover agents, marking targets for it to attack. So if you transplant an organ from one person to another and the recipient's immune system recognizes the donor's HLA as foreign, it'll attack the transplanted tissue. This can lead to severe blood clots, failure of the new organ, and all kinds of painful symptoms that put together amount to transplant rejection. The problem with these HLAs is that they are incredibly diverse with thousands of variants, which makes it very hard to find a donor whose chemistry matches a recipient. Some people spend years on the waiting list, others never get a match at all. But there are a few ways around the problem, and one of them is to skip humans and move to pigs. You might not find this too flattering, but you have a lot in common with a pig. Your immune system functions in a similar way, and your organs mostly do the same things. And pigs are the right size. You wouldn't want a heart valve transplant from a mouse or a beluga whale. The main thing is that a heart valve doesn't need to be alive to do its job. It's really just a piece of tough, flexible connective tissue that acts as a glorified piece of plumbing to direct blood flow. So if you blast the pig valve with a chemical called glutaraldehyde, you can preserve it while killing all of its cells in the process. Said cells can't produce those HLA proteins, so there's much less risk of rejection. The risk is not zero, though. These heart valves can wear out over time since they're dead and unable to replace damaged cells. Also, sometimes the patient's immune system still gets nervous about having this foreign thing in the body. The dead cells still have some antigens on their surface, and patients, especially those with stronger immune systems, can react to them. The valve may eventually fail. So scientists figured out how to minimize even that small risk. They identified the main antigen that our immune systems attack in pig tissue, called galactose alpha-1,3 galactose, or alpha-gal for short. Then they genetically engineered pigs to not express alpha-gal, so the human immune system can accept the pig valve much more readily. But why stop there? Why not use pigs to grow not just heart valves, but whole hearts or anything else you might want? That could solve the organ shortage. Well, scientists are working on it. Knocking out alpha-gal alone is not enough to convince a human body to accept a whole organ from a pig, but some more extensive genetic engineering could help. For example, you could remove the gene that tells the cell to produce some of its other antigens, or throw in some human genes to make the tissue seem more familiar to the human immune system. They've tested these genetically engineered pig organs in non-human primates like baboons, which only survived for a short time, but the technology is advancing quickly. The downsides? Some people aren't comfortable with the idea of genetically engineering pigs to express human genes, even if it's only to trick the human immune system. Plus, cloning enough pig cells for the technique to work is mega expensive. For now, these genetically engineered pig organs would be too costly to reach any patients who aren't like at the private jet level of wealth. There are alternatives in the works, though. One concept hinges on the idea that there is virtually no risk of rejection when an organ is made up of the patient's own cells. So say you take an organ, like a lung, from a human or animal donor and remove all of its cells except the connective tissue that gives the lung its shape. Then you replace those donor cells with stem cells from the patient. Cells love to stick to that connective tissue, so they'd move in and start dividing and differentiating into the proper kinds of cells. This too has been tested in animal models, but researchers still have a ways to go before they can build a completely functional organ. So the science of xenotransplantation still has a long road ahead of it. But eventually the day might come when we will be able to make organs to order, and it will likely be thanks to our porcine friends. Thank you for watching.
So that is xenotransplantation. And like you said, there are still a lot of issues to this and a lot of barriers. Complement-mediated rejection, species-specific MHC proteins on the donor organ are detected by the complement organ of the recipient, the person receiving the organ. When an animal organ is transplanted into a human, the pig's MHC proteins are so different, they trigger an immediate and massive immune response known as hyperacute rejection. This usually destroys the transplanted organ within hours. It can also be rejected by our T cells. Our T cells just don't like it and start attacking it. Recipients usually have to take immunosuppressant drugs for the rest of their lives to suppress their own immune system so their bodies don't reject the donated organ. And even then, our body still sometimes rejects it. Disorders. Mutation of the genes associated with the immune system can cause disorders. Anyone who has allergies is just an overreaction of our immune system to something that is totally common, like pollen or pet hair. A severe allergic reaction is called anaphylaxis. You heard anaphylactic shock. People who are allergic to peanuts and they encounter a peanut sometimes go into anaphylactic shock. It requires an EpiPen, pen full of epinephrine hormone to restart their heart. There's also just abnormal reactions. Your body rejects healthy cells for no apparent reason, and those are autoimmune diseases. Type 1 diabetes is an autoimmune disease. Multiple sclerosis, lupus. Our bodies attack us for no apparent reason. There's also immune system deficiencies. X-linked A gamma globulinemia is one, and severe combined immunodeficiency disease, or SCIDS, is another, which is similar to the viral diseases of HIV and AIDS. Allergic reactions, like I said, it's an overreaction in the immune system. There's an associated video if you'd like to watch that. If you're allergic to anything, it might be interesting to see how your body reacts to those things. When a histamine is released into the circulatory system, it causes a life-threatening decrease in blood pressure and constriction of airway in the lungs. Constriction is the opposite of dilation, where your blood vessels get tighter and then not a lot of blood can get through, or not a lot of air can get through because it's constricting your airways in the lungs. This is what anaphylaxis or anaphylactic shock is. It causes the bronchial tubes to constrict, restricting airflow to the lungs, making breathing difficult. It also causes irregular heartbeats and cardiac shock that can develop in a few minutes if your brain and your tissues don't get enough oxygen because your airways are closed. And SCID is a rare genetic disorder of the immune system caused by a complete absence of both antibody-mediated and cell-mediated immune responses. As you can imagine, you cannot encounter any kind of pathogen if you don't have any kind of immune system. One of the longest known survivors was David, the boy in the bubble, who died at 12 after being isolated in a sterile plastic bubble for all but the last 15 days of his life. And there's a video there if you're interested in his story. He was born without an immune system at all.
Being isolated at home can be rough on a person. And now many of you viewers.
There we go. And HIV and AIDS. AIDS is a collection of disorders that develops after an infection with human immunodeficiency virus, or HIV. It just infects and destroys helper T cells. Approximately 35 million people are infected worldwide. And then I included a website for AIDS statistics for Riverside County if you want to see them.